Welcome, everyone. Okay, as usual, can we make a start, please? As usual, we're going to expect more people trickling in as we go along. We have also, as usual, our online audience. I think there's about 100 people online at the moment. We keep it sort of alive as a hybrid session, but I have to say that hopefully we're not going to see too much of a balance shifting towards really people disappearing out of the audience. So we will keep an eye on that. So I'm very pleased that actually 3 international experts have accepted my invitation to talk to us today about novel treatments for schizophrenia. And before I'm going to introduce the first speaker, I would very briefly like to set the scene almost from a clinical or clinician's perspective. So schizophrenia presents a significant challenge to psychopharmacology. There was a paucity of new drug developments during the last at least 3 decades. Some people would say that even the second generation antipsychotics have not really added much really to the portfolio, given that they predominantly reduce extrapyramidal side effects. But we know now that it came at the price really of lots of people running into metabolic syndromes, which, by the way, also led us as a trust to engage with a multi-center trial, the Pegasus trial, where we are trying to address that as we go forward. Now, 1 in 3 patients is still a non-responder to established antipsychotics, something we clinically forget about. It's defined as no response to 2 trials of antipsychotics, and I think there is a lot to say about the way these sort of treatment resistance categories are established. I don't think we are actually as sort of academically minded around the non-responders as we should be, but hopefully we'll hear some new positive, promising advances today. Efficacy on negative and cognitive symptoms, as you know, is quite limited. I mean, the only substance where they substantial evidence base remains to be clozapine. And I think our understanding of the neurobiology of the potential novel targets is also quite limited. So in that context, the Lancet paper 24 last year asked the question, so is there time for renewed optimism for schizophrenia? Pretty much at the same time acknowledging that even the pharmaceutical industry seems to have to some extent pulled out really of the investigations for novel substances. But the muscarininergic receptor agonists seem to be the substances that create the most excitement. If it does approve, I can't remember, it was I think in late 24 when this was published, if that happened, by the way, but it would be the first antipsychotic for schizophrenia that does not directly block dopamine receptors. So the British Journal of Psychiatry also covering that very topic in this paper, schizophrenia, new treatments soon. And at that point, I would like to now introduce our first speaker, Dr. Edward Chesney. He is an academic clinical lecturer in general psychiatry and addictions at the Maudsley and IOPN. He led actually the first ever study to administer high doses of inhaled cannabis, THC, to patients with schizophrenia. He has a significant role in psychopharmacology studies and clinical trials, including inhaled THC and CBD in healthy volunteers, clinical trials of CBD in psychotic disorders, and others. And he's talking about cannabidiol as a novel treatment in the early phases of psychosis. Over to you. Thank you.
Thank you very much.
Stay here because on the screen.
So I've been studying cannabis, cannabinoids, and CBD for the entirety of my research career, which isn't that long, but it's almost a decade now. And sometimes I worry that I'm drinking the Kool-Aid and through the looking glass and a bit too much of an advocate of them. So I'm quite interested to know what everyone here thinks about CBD. You've certainly heard about it. Lots of your patients probably smoke cannabis or talk about CBD. There's all these CBD supplements you see in shops around the high street. But you all may also be aware that CBD has been approved as a treatment for epilepsy, and that researchers around the world are very interested to see if it can treat psychosis too. So I've got a question for everyone here. What do you think? Who here thinks that in 20 years' time, CBD will be prescribed for people with schizophrenia and psychosis? Okay, there's quite quite a few people. I say I've made about a third of the audience. So we've got a lot of positivity about CBD. What about a different cannabinoid, THC? Who here thinks in 20 years' time we'll be prescribing THC for people with schizophrenia? Nobody at all. Good. You've fallen for my trap, because I actually think that there's more evidence, or more chance at least, that we're going to be prescribing THC. So in the first part of my talk, I'm going to be talking about CBD, and the second half, we're going to talk about THC. So thanks for the introduction, Frank. You had the perfect intro about why we need new medications for schizophrenia. We've only got really one option at the moment, maybe two or three, with clozapine and KarXT. The reason why CBD is interesting is because of relapse and because of adherence. The number one reason our patients with schizophrenia end up in hospital is because they stop taking their meds. And that's because they've got nasty side effects. There's lots of stigma associated with the illness and with the treatment too. Some things we can't change. We're not going to necessarily change insight. But CBD is also such a fashionable, trendy drug that even if you don't think you've got schizophrenia, you may want to take CBD because it's a cool medicine. So what is CBD? So CBD is cannabidiol. It's a cannabinoid. Cannabinoids are these compounds with similar structures from the cannabis plant. There's over 100 of them. The two main ones we all know about are THC, that's the one which gets you high, and CBD, that's the one with the medicinal benefits. And they've got very, very similar structures. They've got the same precursor. It's an interesting question to ask, well, why do plants make cannabinoids? And they make a lot of them. And part of the reason they make so much is because they've been genetically engineered or bred to have so much. You can see here on this, is that point on the screen? That's not. The image at the bottom left, those are trichomes, these little kind of vesicles which bud off the leaves and the flowers containing the cannabinoids. And it's not exactly known what they're there for. They may stop animals from eating the plant because it makes you intoxicated. It's not very nice to be high if you're a giraffe or something. And they also taste bitter. CBD doesn't taste very nice at all. Another thing that's been proposed is they act as a bit of a sunscreen. They're antioxidants which absorb light and free radicals, so it may protect the plant from excess sunlight. What you'll know most about cannabinoids is the kind of CBD health industry and the medicinal cannabis industry. And that's why I put the image of these cans there. You see it all over the place. I'm not sure who buys these, to be honest, because there's no evidence to have any benefit at all. The amount of CBD in these drinks is trivial. But there's also the medicinal cannabis industry, which is getting bigger and bigger, even in the UK now. And there's all this idea that certain CBD to THC ratios in cannabis have different effects. While kind of a high THC blend might be good for getting high, a high CBD blend might have anti-inflammatory effects or be good for pain relief. I think there's one key fact in the kind of drug development story here. You need to know that is that cannabinoids are very lipophilic, very fatty. They don't dissolve in water. So you can see if you've really bred your cannabis, you can make it super shiny. It's dripping with these kind of fatty lard-like cannabinoids. But even got Snoop Dogg there. He's talking about the real sticky, icky. That's the best cannabis, which is the stickiest. If you're a pharmacologist and you want to measure a drug's lipophilicity, the measure is something called the log P, and that's when you get some water and some oil, you give it a shape with the CBD in it, and you see where the CBD goes. The measure for that is how lipophilic it is. The higher the number, the more lipophilic. CBD's log P is 6. Now that's really very high. It's really lipophilic. And if you look at the graph here, the blue bars, these are drugs which have been developed and been approved. Not many have a log P above 6. It's not an ideal medicine. Most pharmaceutical companies would not touch CBD, THC, or other cannabinoids with a barge pole. When you think about the kind of underlying physiology there, it's interesting to notice that these are such lipophilic molecules, because a lot of the mechanism of action of cannabinoids is intracellular. It's not receptors at the cell surface membrane, it's receptors on mitochondria. So the CB1 receptor is found intracellularly on mitochondria, And when THC arrives, or an endocannabinoid, your brain's internal cannabinoids arrive, they tone down the activity of mitochondria in the brain. And this is what underlies the behavioral effects of THC. So it makes sense. We need fatty molecules to get into the cell and have the activity. Because they're so lipophilic, or because CBD is so lipophilic, that makes its pharmacology really complex, and it's a real right pain to study, to be honest. For a start, the pharmacokinetics are really unreliable. The massive food effect, if you take CBD with food, the absorption goes up by 4 or 5 times. People see that when they have cannabis edibles. It can really matter what you've dissolved your THC in, if it's a brownie or something else. You need to dissolve it in something fatty like butter, which is what people do. It also makes studying what it binds to really difficult. CBD is a drug without a target. In a recent systematic review, they described over 65 different receptors and enzymes that CBD binds to. That's not very helpful. Luckily, we can trim it down a bit, because most of these are at super high concentrations, which aren't necessarily physiologically relevant. But then still, if you go down to kind of nanomolar, submicromolar ranges where this is where the effects will be happening, we've still got 5, 6, 7, 8 different candidates. We really don't know how it works at the molecular level. A lot of people question whether it actually binds to everything, partly because there's so much junk science in the field. Because of the role of the medicinal cannabis injury, because so many people believe in the healing properties of cannabinoids, there's some real junk science out there. It's very difficult to line up all this kind of cellular in vitro work with what we see in humans. There's also been some slightly more out there proposals that it acts to adjust membrane fluidity. And so you've got your cell surface membrane with all these, the lipid bilayer. CBD can go in there because it's so lipophilic and adjust the kind of fluidity of it. And that's why at high concentrations, you see that it affects so many different targets. It's also been suggested that it can act as an antioxidant. Lots of antioxidant medications have been trialed in psychosis and schizophrenia. Some have had a bit of an effect, but they've never been that successful. So at the moment, it's a really open field and no one really knows how it works, even if it does. So it's a drug without a target, so that's not very good, but it has got exceptional acceptability and tolerability. I did a survey, I asked 100 patients with psychosis, do you want to take CBD? And even I was surprised, 86% said they would take it. You wouldn't get that with haloperidol or olanzapine. And I think we had about 50 people who had ever used cannabis, and every single one said, yeah, I want to take CBD, doc. So that's encouraging. So people will want to take it. And also once they start taking it, they're not going to stop, hopefully, because it's been described as a placebo-like adverse effect profile. That's not the case. It does have some side effects. It's diarrhea and stomach upset can happen. It's also got interactions with other medications where CYP enzymes. But it's got a very benign side effect profile. Very few people struggle to take it over the long term. So if it does work, it would be fantastic. Even at very high doses, so a CBD supplement's got 10 milligrams. In this study, it's very small on you can see the graph at the bottom there. The writing's very small. That's giving 4.5 grams, only 450 times the dose. Still wasn't really having any subjective effects. So here we go. We've invented a fantastic medicine if you have a good sickness for it. We hope that sickness might be schizophrenia. It's not all, we don't need to be quite so cynical though, as CBD has been approved as a treatment for childhood epilepsy. Now it's 1, 2, 3 different syndromes, Lennox-Gastaut syndrome, Dravet syndrome, and tuberous sclerosis. These are not the most common types of epilepsy, but they do occur, and it does seem to work for them. The doses, again, are about, in an adult, it'd be about 1 gram a day to 2 grams a day. Big doses, far more than you can get from Holland and Barrett. The main issue is that it's very expensive because it naturally derives CBD. There's all these farms out in Kent and Norfolk and Suffolk, I think, growing vast quantities of cannabis, and it has to be refined and purified. It's a very expensive medicine. And as a result, it's only a 2nd, 3rd, sometimes 4th line treatment and prescribed as an adjunct, not as the primary treatment. So the question that we want to now to finally get there is, well, does it work for psychosis? It's an interesting story because a lot of the preliminary evidence came from the 1970s. This paper here, I think, is one of the first randomized trials from 1974 on the left by Carniol, showing that CBD interferes with the effects of THC. And there are around this kind of in the late 70s and early 80s, there are a few more studies like this, but then I think possibly due to legal regulatory changes or just disinterest, the research all kind of stopped. Nothing at all really happened in the 80s or the 90s, but in the early noughties, the research started to pick up again. First of all, with some animal studies showing that CBD inhibits hyperlocomotion induced by psychotomimetic drugs. That's encouraging. And then some observational research. You've got Celia Morgan and Val Curran here on the bottom right. Observational research from UCL showing that people with more CBD, I think it was in their hair, had fewer unusual symptoms. Now, the first big study to come out and get the field really excited was in 2010. This is Paul Morrison on the top right, who led the study. And this is comparing the effects of CBD and THC in healthy volunteers. So we'll see if my YouTube link is working. Oh, it's going to be loud, I think. Bear with me.
But are these views right? Is it really true that the skunk out there can play havoc with your mind? Here at the Institute of Psychiatry, I've agreed to take part in a unique medical trial designed to find out. It's part of their ongoing research into the link between stronger cannabis and psychosis.
That's not very good. You can see that on my screen, but you can't see it up there, can you? Is there a way to fix it? No, that's fine. We'll skip over that video. That's a shame. So what the video showed, that was a journalist there, she got an injection of THC, which she didn't really like. And then the second time she had an injection of CBD before she had the THC, and it was much nicer. And you can see on the graph here on the right, this is looking at psychosis symptoms induced by the THC. The group who had the CBD treatment had fewer psychosis symptoms. This was followed up a few years later by my colleague Amir, who did a similar study using oral CBD, 600 milligrams as a pre-treatment compared to placebo. And again, an IV dose of THC to induce a psychotic reaction. This is in 48 healthy volunteers. And again, psychotic reactions were less likely in the group who had received CBD treatment. So this is pretty exciting. It's about 10, 15 years ago. It's all coming out. Maybe CBD could be used as an antipsychotic. This is when I started to get involved in research, about 10 years ago, back when I was an SHO. And this paper was proposed by Amir and Robin Murray, Philip McGuire, Tom Freeman. They were proposing that, wow, it looks like CBD can reduce the adverse effects of THC, the psychotic effects, paranoia-inducing effects of THC. Perhaps if we add a bit of CBD back into recreational cannabis, it will make it safer. So we designed this big study. We got 46 healthy volunteers, and each person had to do 4 different sessions, 4 different blends of cannabis. Every time, each blend of cannabis always had 10 milligrams of THC. That's a big dose. That's more than enough, thank you very much. And then increasing amounts of CBD from 0 to 10 to 20 to 30 milligrams. This kind of covers the whole range of recreational cannabis. The doses are lower than the oral doses, but much more gets absorbed when you inhale it. So we were hoping that the more CBD we put in there, the less paranoia and psychosis you'd get. Unfortunately, here are the results. You got, ignore the gray bar, that's the baseline, but the 4 different color bars there are the 4 different arms. There was absolutely no difference in any outcome at all. All types of cannabis are exactly the same. All that matters is how much THC you're inhaling. It's not all doom and gloom, because there has been a small phase 2 trial. This is Tom Freeman, who when he used to be at UCL, he's now at Bath. He did a small phase 2 trial giving CBD to people with cannabis addiction, cannabis use disorder, and showed that treatment of CBD will people get off cannabis, help people reduce their use. And a phase 3 trial is now underway. The main part of evidence for CBD is the clinical trials, of which there have now been 4 as of last year. The first one was from Germany in 2012. And they randomized 42 people to treatment with CBD 800 milligrams. This is as a monotherapy by itself compared to amisulpride, a very effective antipsychotic medication. And at the end of treatment, there was absolutely no difference. Well, there's no statistically significant difference in psychosis symptoms, but fewer adverse effects in the CBD arm. So that is really encouraging. CBD in this trial was as good as amisulpride. There's no placebo arm. People would still want to see that because it could just be a regression to the mean. People's illnesses are resolving naturally anyway. It's very encouraging to see that. Next came out a study from Yale, where they used a lower dose of CBD, 600 milligrams, and the formulation they used wasn't that good. This time they're prescribing it as an adjunct on top of antipsychotic medication, which makes it harder to show an effect. And unfortunately, they found no effect at all. The best study which has come out was by my colleague Philip McGuire, professor in Oxford, comparing 1,000 milligrams of CBD. This is Epidiolex, the epilepsy medication, so good dose for 6 weeks of treatment as an adjunct to antipsychotics. This is again in people with schizophrenia, and showed that the CBD group, it reduced psychosis symptoms and improved overall clinical impression. And again, very few side effects. So that's 2 out of 3. Just last year was published a paper by Sagnik Bhattacharya, researcher at King's. This is a smaller, shorter trial, just 33 people getting treatment for 3 weeks, but it was the prodromal psychosis group, the clinical high-risk group. This is really exciting and really interesting because if we can prevent people getting schizophrenia, that would be fantastic. This was just a small study, so it wasn't looking at prevention, it was just looking at kind of prodromal psychosis symptoms, but it did find the CBD group had fewer prodromal psychosis symptoms at the end of treatment. So we've now got 3 out of 4 studies with encouraging evidence, which sounds great, but if you look at the history of clinical trials, there's always lots of phase 2 trials which show amazing positive effects, and then you go to phase 3 and everything falls apart. And that's where we're moving towards now. So I'm involved in the STEP trials. This is together is 3 clinical trials, 1,000 people in total who are going to be given treatment of CBD or placebo. We're going to look at 3 different populations. One's the clinical high-risk group, and they're going to be offered treatment for 2 years. And then we want to see if we can actually reduce people developing psychosis. We're also going to give it to people with first episode psychosis who haven't responded to antipsychotics, and people who've been given clozapine, but despite clozapine still have symptoms. So these 3 trials are going to be recruiting from this year all across Europe and Israel. And maybe in a few years' time, we'll know whether or not this was all worth our efforts. So to summarize, CBD, it's got wonderfully high acceptability and tolerability and preliminary evidence of efficacy. If it does work, and it might, I think there's a reasonable chance, it would be a fantastic long-term treatment. It's a medication people want to take. It doesn't have too many side effects. It'd be really good for relapse prevention. It'd be really good for the clinical high-risk group too. And because of the side effects, it'd be good for younger people who experience more side effects, for people with first episode psychosis too. Again, we don't know its mechanism of action, but it's not dopamine antagonism, so it might be good for people with treatment resistance. And it might also be good for people, for cannabis users, if it has the additional protective effect in that group. One last point I'll make, if anyone here still recommends that people smoke cannabis with high CBD in it, it's not wrong, because the more CBD a cannabis plant has in it, the less THC will have in it, but the CBD doesn't really matter. What people should do is just smoke less THC. Okay, so now I'm going to move on to THC. So let's drink first. So everyone knows about THC. That's the stuff that gets you high, and it's responsible, and it's a partial agonist at the CB1 receptor. CB1 receptors are all over the place. I didn't ever learn anything about CB receptor when I was at medical school, but they're one of the most prevalent receptors in the brain, all over neurons and astrocytes, and the ones which seem to matter the most are the ones in mitochondria, because you can selectively eliminate the ones on the cell surface membrane or the mitochondria and see how that affects mice behavior, and all the effects on mouse behavior, all the effects of cannabis are mediated by the mitochondrial ones. It's addictive. It's not the most addictive drug, but it's probably as addictive as alcohol is. I think at least 20% of people who smoke cannabis end up having some sort of problem with it. And teenage cannabis use is probably one of the most important risk factors for the development of psychosis. And some have claimed that a quarter of cases in London are accounted for by cannabis use. A bit of cannabis is fine. Most people have a nice time. It's relaxing. It's enjoyable. But overdose can cause acute and transient psychotic symptoms. So why on earth do I think that maybe in 10 years' time, people in this room will be prescribing THC for people with schizophrenia? That's because of cannabis withdrawal syndrome, which I think is something which people don't diagnose very much, and I think they should. It wasn't recognized in DSM-4. It was only after lots of research in the 20 years ago that it was officially recognized in DSM-5. That's because people don't think it's real, or didn't used to think it was real. If you survey people who are heavy cannabis users, only 17% say they experience withdrawal symptoms. But then if you bring them into the lab and stop them smoking, 87% are observed to have it. And 50% of them complain that the symptoms are severe. So it's definitely present. Why is it under-recognized? I think because the symptoms are subtle, relatively subtle. They're more emotional, behavioral, they're less physical and somatic symptoms. You do get some somatic symptoms, like you can get abdominal pain, sweating and shakes. But the main ones are irritability, insomnia, anxiety, and feeling restless. Interestingly, these are often the reasons people smoke cannabis. It's the indications for medicinal cannabis. So people think, oh, I've got insomnia, I need to smoke weed every night. Well, maybe you've actually got cannabis withdrawal syndrome when you don't smoke your weed. Compared to other drug withdrawal symptoms, it's also got a slower kind of onset. If someone doesn't drink alcohol, someone alcohol dependent and stops, they could have a seizure within 12 hours. It takes 3 to 5 days for the symptoms of cannabis withdrawal syndrome to become obvious, but they can be quite obvious eventually. And just mention, obviously, most people use cannabis with tobacco, and the two withdrawal syndromes combine to make things worse. I'm going to describe a case report. Back when I was doing my PhD, I did a lot of Section 12 work, and that was good fun. And one night, that's 2 in the morning, I was at St. Thomas's Hospital and I was asked to see a young woman. The story was that she'd gone off to university and done what lots of young women do, which is smoke loads of weed. That's all fine. But she got in an argument with her boyfriend. Maybe she was showing prodromal symptoms. He wasn't happy that she was smoking so much weed. So they had a big argument, they broke up, and she wanted to get back. So she threw all her cannabis in the bin. The next night, she couldn't sleep. She couldn't sleep. The night after that, or for the third, fourth, or fifth nights. Eventually, things started to go wrong, and her family drag her back to London, and they're very concerned about her because she's paranoid and hearing voices. This got me thinking, that makes sense. If you can't sleep for 5 days, I think there were studies from the 1950s of US Army soldiers not sleeping for a week, and they would develop psychosis or psychotic symptoms. No wonder that severe cannabis withdrawal syndrome can trigger a psychotic episode too. So we did 2 studies. One was a systematic review of previous case reports. This has been described in the literature only 40 times ever, so it's not very common. So I was a bit disappointed. But then I did my own more systematic study. We searched through the case notes across all of the South London and Maudsley Trust, which is 4 boroughs of London. And by searching for terms, cannabis withdrawal and psychosis, if those appeared in the same note, I would review it. I was able to find 68 cases, which again, isn't that many. Then only a third of these cases did the clinician clock that cannabis withdrawal was occurring. They said, stop smoking weed a week ago. They said, not been sleeping for days. And they said psychosis, but they didn't kind of tie it all up. So I think there's actually way more of this happening out there. I don't know how common it is, but I think it does definitely occur. And I wonder if there's a bit more awareness of cannabis withdrawal, people might diagnose it more often. The type of case was very consistent. They were all heavy cannabis users, at least using a gram a day. That's loads. They were all stopping suddenly. Most of them were male, and over 90% there was a report of insomnia, and lots of people were not having a wink of sleep for several days. This is the second study. So this one's been published in the BJ site just last month. This is the second study I've done. And it kind of occurred to me, wait a second, half my patients are dependent on cannabis. When we admit them to hospital, we're going to force them to stop smoking weed. So they're going to develop kind of iatrogenic cannabis withdrawal. So I designed a study where we identified every single patient who'd ever been admitted to the Maudsley Trust. I think there are tens of thousands of admissions. But we identified the subgroup who'd been not initially admitted to intensive care, but transferred after initially admitted being to a general ward, kind of indicating that they'd been okay, but things have got worse during their admission. And we set the window, the risk period is days 3 to 5. That's the peak of cannabis withdrawal syndrome. And you can see here on the graph that there is a little jump in the proportion of cannabis users who are transferred during that window, 31% as opposed to 24%. Now, this isn't a massive effect, but it is statistically there. It has occurred. And also just recognize that the data I'm using here is crap, basically. It's clinical notes. Does the clinician think they're a cannabis smoker? They might be right, they might be wrong, but there's a lot of noise in here. So it's quite exciting that we found something. What's more interesting, it adds to the interest here, is looking at subgroups. Oh, yeah. In women, there's a massive big increase, and in older people. For some reason or another, women develop cannabis addiction much more quickly than men. There's also interesting interactions with the menstrual cycle and between the endocannabinoid system and estrogen. So it's possible that it could be worse in women, and also in older patients. I think this is just a group this is the group who've kept smoking cannabis for many, many years, and they're never, ever going to give up. It's just a marker of severity. I think from these two graphs, I think it's quite clear that there is an effect here. This brings us to the Sativex trial, which is happening down in South London. This is so ideally, I wonder in the future if we'll be prescribing, just like we do for people with alcohol dependence, we give them chlorodiazepam epoxide or diazepam. We might give them a bit of THC to mellow them out over the first few days of their admission. Whether you want to do that in someone with acute psychosis is an interesting question. At a ward in South London, they're not quite doing that, but they're getting people on the ward who've got psychosis and a cannabis use disorder. And after they've been stabilized on the ward, after the cannabis withdrawal syndrome is settled too, they're offered low to medium doses of Sativex. That's the spray which is approved as a treatment for multiple sclerosis. The aim isn't to treat the cannabis withdrawal syndrome, it's to stop people when they're given unescorted leave to going out and celebrating with a massive joint. The clinicians come to a deal with them saying, if we give you a bit of weed a couple of times a day, will you play the game? It's only been done in about a dozen patients so far, but it seems to be working well. This is, I think the treating cannabis withdrawal syndrome is a concern safety-wise, but it might not be quite as controversial kind of politically as this one. But it's interesting to see where this field could go one day. So to summarize what I think about THC, first of all, cannabis withdrawal syndrome is very common. 87% of people experience it. And I think clinicians, we're not taught to look for it. If you do want to look for it, get the cannabis withdrawal scale out. It's really easy to use. It got all the symptoms there. Patients find it really useful to go with them. Go for it. It's often pretty severe, 50%. I think it can trigger a first episode psychosis. I'm not saying it's the cause. These people are probably going to get psychosis one day anyway, but it can be that thing which just causes the initial episode or the relapse. And it's probably a significant cause of violence and agitation in inpatients. There is an easy solution available, and that's THC. It just might be a bit controversial. And it's got very interesting implications for our approach to management of cannabis use disorder. I think lots of things like insomnia, psychosis, EPD, anxiety disorders are complicated by cannabis use disorder at best, and some of them may just have lots of underlying cannabis withdrawal symptoms. I've started in my clinical practice to teach my patients about cannabis withdrawal symptoms, and it's been really quite impressive, actually. I think that if you're going to take away one thing today, it's that you can psychoeducate your patients. I often previously used to be really stuck with cannabis users with psychosis, and there's not much you can do with them. They're never going to listen to you. But perhaps if you sit down with people and discuss cannabis withdrawal syndrome, they might look at things in a slightly different light. And hopefully this will be the future. Gosh, it makes so much sense. I must slowly reduce my intake over the coming weeks. In the meantime, you can offer symptomatic management, benzos for the insomnia. Sometimes I think people need something stronger like quetiapine or olanzapine to get to sleep if they're going to stop smoking weed. Paracetamol for the headache, hyoscine for the abdominal upset, and metoclopramide for the nausea and vomiting. So there's lots you can do for this, and I think it's the smallest lesson people could take away from today. So to summarize my talk, CBD has many strengths. It's got extremely high acceptability to patients. 86% want to take it. The side effects are pretty minimal. It's very well tolerated, and it's probably got a novel mechanism of action. We're just not sure yet if it's actually going to work. You're going to have to hold tight for another 5 or 10 years. THC, on the other hand, has lots of weaknesses. Who here wants to prescribe it to vulnerable populations of acute psychosis? I expect not many people. Definitely got side effects. High doses will cause psychosis and paranoia. Interesting, it will definitely be acceptable to the patients. They love it. Whether it's going to be acceptable to staff and the public, we'll see. Are we encouraging cannabis use? Or perhaps are we building therapeutic alliance? It's not particularly controversial to give opioid agonists to people with opioid use disorder or benzodiazepines to people with alcohol use. Why can't we give CB1 receptor agonists to people with cannabis use disorder? It has all these limitations, all these interesting questions, but I'll say the efficacy is pretty much certain for cannabis withdrawal syndrome. We know why they've developed it. It's the lack of THC. We just need to put some back there. Well, I'll finish there. At the start, I did a bit of a poll asking people if they think CBD is going to be prescribed or THC is going to be prescribed. I think we had maybe a third of people were keen for CBD and no one for THC. So in your career, which one do you think you're going to prescribe for acute psychosis first? Who put your hand up now if you think it's going to be CBD? A few people. Who thinks they're going to prescribe THC first? Not many. So, okay. There may be some converts. Thank you very much. And I'll finish there. Thank you very much.
Thank you. And we're moving even further with aspects really of genetic relevance in terms of the very question, how can genomics help find new treatments for psychosis? So something the other way around, I suppose. I would like to introduce Professor Paul Harrison, who is Professor of Psychiatry, the Associate Head of Research Department at the University of Oxford. He's also the Chair of the Oxford Neuroscience Committee, Honorary Consultant Psychiatrist, although just stopping to work clinically, as I just heard, at the Oxford NHS Foundation Trust. His research addresses several aspects of translational psychiatric neuroscience, and on his website he wrote, "I have a particular interest in the risk genes, which represents potential treatment targets such as voltage-gated calcium channels." So looking forward to your talk. Thank you.
Yeah, thank you very much, Frank. Thank you for the invitation to take part. Yes, so I'm a psychiatrist from Oxford, and if you're like the psychiatrists I talk to in Oxford, you're not all molecular biologists or geneticists. So apologies if you are, but I'm going to give this a fairly kind of high-level introduction. Now, as we heard from Frank and indeed from Ed, we haven't really had substantive new treatments for psychosis or indeed for other psychiatric disorders for at least 50 years. We've tweaked the same mechanism and come up with some slightly different drugs, but really nothing has made things any better. And that's kind of, it's a vicious circle. It's partly because we don't understand the pathophysiology of the conditions that we're trying to find drug treatments for, but because we don't have the treatments, it's hard to know where to start your research if you're looking for new pharmacology. And people have held out the hope that genetics might sort of break the impasse. And you'll all be aware that there's been a massive funding of and carrying out of genetics research in psychiatry for the last 25 or 30 years, and substantial progress really in the last 10 to 15 years. And although I'm going to talk specifically about schizophrenia today, almost everything I say in principle would apply if you're interested in ADHD or depression or any other condition. Because for all these conditions, we've found quite a lot about the genetic basis of these conditions. We've identified quite a lot of the genes. And the question is, for today, to what extent does that help us identify new treatments that are getting closer to the cause of the illness? So I'm going to run through initially just a very brief introduction to kind of how genetics can help find new drug targets, a very brief overview of the genetics of schizophrenia, and then take a deep dive into one of the particular candidate pathways that we and others are interested in building upon this genomic information. So why bother finding genes? Well, if you're like me, it's just interesting. And I happen to believe that if you understand the genes that contribute to a disorder, you'll understand the pathophysiology. And that in turn is interesting both in itself, but ultimately leads to clinical benefits. And the first of those benefits could, of course, be in diagnosis. It is possible that when we know more about the genetics of schizophrenia, we will use genetic markers to define subtypes of the illness. It's one approach to personalized psychiatry. And there are other areas of, certainly of neurology and medicine, where that's proven to be the case. It may also be that genetics helps us with understanding prognosis. There's a little bit of evidence that certain genetic profiles predispose you to a more treatment-resistant form of psychosis than a more treatment-responsive one. But last and the most relevant today, can it really help us get to therapeutics? And the evidence in the rest of medicine is yes. So if you look at drugs across the whole of medical specialties, a drug that happens to hit a target that is part of the genetic background of that condition, the chance of the drug working are about twice as good as if there's no genetic support for that target. There are a few rare, more striking examples, but that's the overall picture. So knowing about genes increases your chances that your drug is going to work, but it by no means makes it certain. There's still more drugs that fail that have gone after a genetic target than succeed. But nevertheless, if we've factored genetics into our searches for new targets and drugs in psychiatry, it ought to help. So what do we know about the genetics of schizophrenia particularly? Well, we know that it's one of the more heritable psychiatric conditions. So depending how you measure it, you get estimates of about 60 to 80% of whatever's causing schizophrenia in the population is somehow in our genomes. It's in some mysterious way to do with our genes. Doesn't tell you how many genes there are, doesn't tell you how they're working, doesn't tell you if everyone's the same, but that tells you that there's got to be a significant genetic contribution. And then there's three sorts of genetic involvement. The majority of the heritability that we can now explain comes from these things called GWAS studies, genome-wide association studies, which find these common variants that all of us have throughout our genomes, but particular ones, if you happen to have variant A, not variant B, they're also called polymorphisms or SNPs, you're a bit more likely to get schizophrenia than if you've got the other variant you could have had at that place in the genome. I'll come back to a slide about GWAS in a moment. But then there are also things called copy number variants, and this is where you have a chunk of your DNA missing. So instead of a single nucleotide being different between individuals, it turns out we've all got pieces of DNA from chromosomes missing. And when I say a piece, I mean maybe 1 million nucleotides in a row from one of each pair of the chromosomes that we have. And there are certain pieces of DNA you can have missing, CNVs, which then significantly increase your risk of schizophrenia because, presumably, the chunk you're missing is a chunk of DNA that has a gene that you need or that protects you from other harmful outcomes. And CNVs are now, in a recent publication of the Royal College you may have seen, they're now recommending we should be thinking about testing for CNVs in particular in people presenting with psychosis, because the diagnostic yield is enough that you may pick up a cause in that patient. Now that's a talk for another day. There's all sorts of ethical as well as practical issues to do with that, but that's an example of the genetics is getting closest to clinical practice. To complete the picture, there are things called rare variants, and they're really very rare variants, which are a bit like polymorphisms in that they affect a single base in the DNA. But if you have that variant, it has a big effect on the protein that that gene is encoded. They're almost like mutations. They're not quite, but they're very, very pathogenic. So you've got these three kinds of genetic difference, which together we think explains all of the genetic basis of schizophrenia. And that's an example, first of all, of what a GWAS study looks like. These are the so-called Manhattan plots. Many people have seen these before, where you're plotting the chromosomes across the x-axis from the number 1 to the sex chromosomes. And up the y-axis is the probability that at that point in the genome, there is a gene or a locus, an area that's associated with schizophrenia. And the higher the peak, it's not the gene is more important there, it's just the more certain people are that there is something to be found there. And if you add up the number of green diamonds, it's about 287. So there's at least 287 genes or loci that are contributing something to do with schizophrenia, and probably many more. Okay, so these are huge numbers of genes, and each of them on their own has a very small effect. So they're only changing your risk of schizophrenia by maybe 5%, 10% at most. Okay? So at a population level, they're crucial. At an individual level, individually, they're of very little significance. And that, of course, is relevant to the treatment opportunities, because if a given target is only contributing a bit of the variant, then even if your drug was fantastic at correcting whatever that genetic difference was, would that really make a big difference to the pathophysiology altogether? If you put together the common variants, the CNVs, and the rare variants together, you get a picture like this, which is sometimes called the allelic spectrum or the allelic architecture of schizophrenia. And here what the y-axis is plotting is the significance of that difference. So an odds ratio of 100 means that if you happen to have that deletion, that CNV on chromosome 3, you're about 100 times likely to get schizophrenia, according to these data, than if you don't have it. You then coming across the x-axis is how common is that genetic difference in the population as a whole. And so all the common variants that give you the Manhattan plots from GWAS studies, they're very, very common, but they really don't make very much of a difference in that individual. So you have this spectrum of things that at one end are really important to the person, but not important in the population, and the other sort of variants are the other way around. Okay? And that's relevant to today because one of the many decisions you have to make when you're deciding which gene you're going after is, do I want to go after something which there's only going to be a few people who might benefit from it, but I really might be able to help that person, from a drug which is going to help lots of people, but maybe not very much. And that's an open, to my understanding, that's an open question. So those are, if you like, the genetic, that's the very brief overview of the genetic picture of schizophrenia. And I said there are hundreds, if not thousands, of genes that have been identified. So how are you going to pick from this huge range the very small numbers that you could possibly have the time and the money to investigate in the necessary detail to go from the potential value of a genetic finding to actually having a drug to test in the clinic? Well, the first thing is the statistical evidence. How certain am I that the gene I'm interested in is not going to disappear in the next study? Is it really robust? So you take a fairly standard statistical approach to that. In those Manhattan plots, many of the genetic signals actually aren't within a particular gene. Lots of our DNA is so-called non-coding. You have a gene here, then a gap of DNA, which we don't know what it does, but may not do anything, then another gene. A lot of the genetic signals are in between two genes. So immediately you've got a problem knowing which gene I think is the relevant one. So one of the things you can use is say, I'll only take the genetic signals that are within a gene, because then it's much more likely that the genetic signal is telling you about what that gene is doing. And you can also say, well, prioritize genes where there's evidence both from the GWAS studies and the rare variant studies, because then you might, if you're lucky, get the best of both worlds. You can also say, let's take a gene that makes sense biologically. The first genome-wide significant gene, schizophrenia, was a gene nobody had ever heard of. It had a series of letters and numbers. People didn't even know if it was in the brain, didn't know what it did. But that was the only gene that had the statistical criterion. So people, including us, went off chasing that wild goose chase, I have to say, which ended up absolutely nowhere therapeutically. Now we've got plenty. So you can prioritize genes that are in pathways where we have at least some pre-existing biochemical or pharmacological evidence that there's something to do with what we think is the pathophysiology of schizophrenia. A lot of the genetic signals for schizophrenia are also genetic signals for other disorders, particularly bipolar disorder, but also unipolar depression, ADHD, learning disability, and so on. So you might say, well, I'll at least take a gene which I know is involved in several disorders because it increases the chances I'll find something that might be therapeutically useful in at least one of those conditions. And then lastly, this thing about druggability. Lots of genes aren't predicted to make a protein that there's any realistic chance of being able to use a drug to target. If, for example, it's stuck in the middle of a cell, if it's not on the outside of a cell, although you can get drugs in, like cannabidiol and THC clearly do, it's not a great start. Or it might be a gene that we think is so fundamental to other aspects of biology, anything I did to use it as a potential schizophrenia treatment might have such awful side effects or it'll be toxic and so on. So there are various criteria by which you can identify genes that, at least in theory, would be druggable. Anyway, as a result of those kinds of considerations, which researchers spend a lot of time mulling over, there's a series of genes that people have sort of kicked off looking at as treatment targets of schizophrenia. And I'm going to leave the first three. And in a moment, I'm just going to drill more deeply into this particular gene family called the voltage-gated calcium channels. In particular, one of those called CACNA1C. I'm afraid the terminology here is a bit annoying. CACNA1C is what you call it when you're talking about the gene, which tends to be what I default to calling it. But the protein it makes is called CAV1.2. So apologies if some of my slides say CAV1.2 and some say CACNA1C. It's the same thing. Okay, so let me just introduce you now to voltage-gated calcium channels, a bit about, again, the rationale for studying them, and what is the evidence at the moment that they're treatment targets for psychosis. Well, they tick the statistical robustness box. So they're unequivocally something to do with genetic risk for at least those three major psychiatric disorders. And crucially, coming back to my druggability point, we know they make drug targets because they are the target of the calcium channel blocking drugs, which I think are the 5th most widely used drugs in the world. The drugs used for hypertension, drugs like amlodipine, verapamil. So drugs that were developed for the cardiovascular system. So we know they can be targeted. We also know from a lot of pre-existing neurobiology that VGCCs play key functions in the brain. I'll come back a bit more in a moment. So therefore, for those reasons, they are at the top of the attractive list of candidates that you might take into study. And the fact that we know so much relatively about these genes and calcium metabolism and calcium channel blockers you've got something to build upon. You're not starting from scratch, as you would be the case for many other genes. So I've just got a few more slides to introduce different aspects of the calcium channels, and then I'll come on to the treatment itself. So this is a cartoon of what a calcium channel looks like. And at the top, it's sort of a two-dimensional spreading out of the protein as it winds its way across the cell membrane, and up is outside, obviously is inside the cell. And it has these different domains. And the main sort of black and grey squiggle is the key subunit. It's the one that makes the channel the calcium is going to go through, and that's called the alpha-1 subunit. But in order to work, you need a beta subunit, I've shown in green, and this alpha-2 delta subunit that's at the top right-hand corner. And then in the bottom of the slide, you can see what it looks like in a three-dimensional formation. So when I say a voltage-gated calcium channel, I mean that combination of subunits, and each of them is encoded by a different gene. And there are a number of genes in this family. And I don't worry about the names of everything, but the pore forming the main subunit, the black and white grey squiggle, you can see there's about 10 of them. Okay, and the cacno1C I've started off with just happens to be one of those 10. There are then some beta subunit genes and some alpha-2 delta subunit genes. In the right-hand two columns are what is the statistical evidence that that particular VGCC gene is associated with a psychiatric disorder. And so you've got the common variants, that's the GWAS plots, and the rare variants. And anything in bold is significant in the latest, basically, meta-analyses of genomics. So you can see a number of these calcium channels, not just cacno1C, but particularly cacno1C, meet the criterion of having something to do with these disorders. Okay. Just before I move on, this is an example of what you get in one of the meta-analyses, where here people have simply taken one of the genetic markers within the middle of this cacno1C gene and said, is there a difference in the frequency of this particular variant in patients with schizophrenia and the controls in different populations and different studies? And pretty much everyone is to the right of the line of no difference. And the meta-analysis, I think, ends up with an odds ratio of about 1.12. So this particular variant in this particular gene affects your risk of schizophrenia by about that much. But that's what we mean by genetic evidence for a common variant being linked to schizophrenia. Just to say a little bit more about the roles of VGCCs. They're essentially all excitable cells. So they're one of the things that when you have a depolarization going down your neuron, the depolarization membrane causes calcium to flood into the cell, and that triggers a series of other events, including propagation of the potential along that neuron. But it also sets off a number of more chronic events to do with synaptic plasticity. So these genes are very important in brain plasticity and adapting to environmental changes. And those are the kind of processes that people think may well be awry in psychosis, and therefore calcium channels are at least superficially doing the right kind of thing to be of interest in that regard. The different calcium channel genes, as I mentioned, have different individual properties. They're expressed more in certain areas than others, but they're all roughly doing the same job. As I've said, their primary interest was in their role in the cardiovascular system. They're abundantly expressed in the heart muscle, and particularly in our arteries, which is why they're good targets for hypertensive treatments. The fact they are expressed peripherally as well as centrally is both a good thing and a bad thing if you want to use them as drugs for a psychiatric indication. It's a good thing in that we've got all this information about what they do and they're druggable, but they're a bad thing in that if you think, well, if I want to target them in the brain, aren't I bound to get side effects in the cardiovascular system? And secondly, if they were going to be used psychiatrically, wouldn't we know that already from the fact they'd be widely used in hypertension? So let me first of all take that, because there's really two ways you can explore these channels as drug targets. The first is, what is the evidence from existing calcium channel blockers used in patients for cardiovascular disease? Does that do anything to their mental health, their brain health? And then secondly, how can we try and tease apart the channels in the brain from the channels in the heart and the arteries? Okay. So let me first of all run through the evidence from the existing calcium channel blockers in clinical practice. What, if anything, does that do to our mental health? Perhaps before I come to that, when the calcium channel blockers were introduced in the 1980s and 90s, people thought, as they always do, let's have a go in psychiatric illness. Do these drugs do anything in mania in particular or in schizophrenia? And there are a number of small clinical trials and case series in the 80s and 90s, which, as often happens, claimed there were some benefits. In particular, people with mood instability who were given verapamil were said to do better than patients who weren't. But when we did a systematic review of this a few years ago, there really wasn't enough evidence either way. So if you take simply clinical trials evidence, we have no idea if these drugs work for any psychiatric indication. We don't. Okay. But what you can do, as I say, is look at people who are taking these drugs for other reasons, so-called pharmacoepidemiology. And the first good study of this kind from Joe Hayes at UCL looked at people in this big Swedish population database with psychosis who were taking calcium channel blockers at one period and then weren't. And if you looked at the periods when they were taking these drugs, they had significantly fewer admissions for psychosis or self-harm than the periods when they weren't taking these drugs. Okay, just a hint that might, there's all sorts of confounders of that, but just a hint, it might be a benefit. And we've done a series of studies looking at a very big American database at over, it's now about 80 million patients, where you simply ask the question, in patients who are taking these drugs and you follow them up for 1, 2, or 5 years, do they have more or less psychiatric events, either first episodes or recurrences, than otherwise matched patients who are not taking calcium channel blockers? So you can match your patient for age, sex, ethnicity, BMI, etc., and simply try and ask the question. And what those studies show really quite robustly is that people taking calcium channel blockers, compared to at least to diuretics and beta blockers, have both reduced onset of and recurrence of mood, anxiety, and psychotic disorder, and a reduced risk of dementia. Okay, I was quite struck by those findings. In fact, other people sort of replicated them in other data sets. But that's taking all calcium channel blockers. And we know, in fact, that not all of them get into the brain. And the most commonly used calcium channel blocker these days, amlodipine, doesn't really get into the brain much. But some of the other new drugs, the other odipines, do. So in a later study, we then said, okay, what happens if you compare people on amlodipine with people taking the other calcium channel blockers? And as you can see in the figure, if you're taking one that gets into the brain, your brain health on those diagnoses is, or you're less likely to get those diagnoses than if we're taking one of the other calcium channel blockers. Okay, so in a way, that's to me the more interesting finding, because it implies that any benefits, if this is true, any benefits aren't just because you're controlling your blood pressure better and therefore having better vascular health. It might be something to do with the fact you're also blocking the calcium channels in your brain as well as the ones in your cardiovascular system. Okay? So big data is quite helpful, again, in giving us a bit more reason to think there might be something in this, but it is only association. There's various residual confounders, which means you can't take this as I don't need to do a clinical trial, this drugs are going to work, I'm going to give it to my patients. This is simply another reason to get interested. Okay. The other approach you can take is experimental medicine. So we did a study where we took volunteers with mood instability and we randomized them to either nimodipine, one of the other calcium channel blockers, or placebo, all matched. And we did an intensive series of investigations in them, including MRI scans, MEG scans, neuropsychology, the works, over a 4-week period. Okay, these were healthy young adults, not hypertensive, just healthy adults. You can see we published the protocol in 2019, and then we had the pandemic. So I'm afraid even 5 years later, we finished the study, we're now analysing it. But that's just an example of another approach you can do to say, if I, in a very controlled way, put people on a calcium channel blocker, and this is one of the ones that does get into the brain, does it do anything to the brain or behaviour? Okay, it certainly does something, but we're not quite sure exactly what yet. So these kinds of studies give us some indication that existing calcium channel blockers have some effects, but they're never going to be indications. They're never going to be psychiatric drugs because of what they're doing in the periphery. The question is, could we do better and get more brain-targeted ones? Okay. To do that, and just jump to the bottom bullet point, we need more efficacy, fewer cardiovascular side effects. You've got to ensure they get into the brain, and not just get into the brain, but ideally sit on the calcium channels that you're trying to block. And how are you going to do that, given that these channels are at least as important and abundant in the periphery? So you're going to have to find something that's different about the brain calcium channels from the heart and aorta ones. Unfortunately, that's where this process called splicing comes in. And some of you may remember this, some of you won't. Alternative splicing is a process by which one gene can give rise to different proteins depending on which bits of the gene information are retained and which are cut out or spliced out. So one gene can give rise to different isoforms. And we know from many other genes that an isoform of the same gene can be very different from its partner in terms of its function, its distribution, and its ability to be blocked or activated by drugs, depending on which domain, which part of the protein gets left in or taken. Okay. So we've taken, and for a reason I'll come back to later, we've taken the approach of, are there isoforms of these calcium channels which are different in the brain than the heart? And almost all the existing data are, first of all, as you might expect, from rodents, not from humans, because human brain splicing studies are more difficult for lots of reasons. And secondly, they come from the heart, because that's what people thought was the interesting organ. So we've now been doing these studies in human brain rather than rat heart. Okay. And we know that species and different tissues make a difference to splicing for many other genes too. The problem is, it's a very large gene, as I'll come back to in a moment, and it's complicated. However, let me just run through a couple of slides showing you what I mean by splicing to make sure we're on the same page. This is a typical gene at the top. You have a thing called the promoter, which kind of regulates the gene. The gene then starts, but you have these, what I've shown here are 4 exons, 4 bits that make the protein, and in between them are bits of intron which don't make protein, and they're going to get spliced out when the gene is being turned into protein. But you can keep any or all of the 4 exons in this gene. So you can have the protein that uses all the gene, you can have one that uses out number 3, you can miss out 1 with 1. So you get a combinatorial potential number of isoforms from each gene. And they turn into proteins of different lengths, which may have different properties. Okay, so that's what I mean by an isoform. And we know that splicing can be regulated by genetics. And there's reasons to think that many of those polymorphisms in the schizophrenia genes, what they're doing is they're regulating how that gene is being spliced. They're not causing an abnormal protein, they're just causing changes in the abundance or proportions of different isoforms in that gene. And that is where the functional consequences come from. Okay, so that's isoforms or splicing in a nutshell. The reality, as I say, is much more complicated. So there's all sorts of ways you can splice in and out bits of genes, and there's all sorts of ways in which genetics can affect that process. So it's an easy thing to say, we're interested in the splicing of Catenin-1C, but the doing of it, of course, is very difficult because this gene has got 52 exons. Now, I don't know how many potential isoforms you can make from leaving in or out any number of 52 exons, but it's a heck of a lot. Okay. We also know that lots of different genetic variants in the gene can regulate these decisions. The blue arrow at the bottom is pointing to the hot spot of the genetic association in the Catenin-1C gene. That's where the genetic signal on the Manhattan plot is coming from. It's right in the middle of the gene, right in the middle of an intron between exon 3 and exon 4. So one possibility is what that genetic variant is doing is affecting which exons on each side of that gene are being left in to make protein or are being taken out or combined in other ways. Now I'm back to a cartoon of the protein, and all I've done here is that you may not be able to read the numbers. The numbers are the exons in the gene giving rise to the different bits of the protein, starting from the first exon at this end down to the 50th exon at the other end. So when I say you could have a calcium channel without the bit made by exon 17, you'll end up with a protein where the bit on the end of exon 16 is joined onto the exon 18. Okay, and I'll show you a picture later about what actually happens. That's the idea. The exons in the gene, which are always used in the same numerical order, even if you leave some out, give rise to a protein with bits of the protein missing that then changes the function of the protein. And identifying that has been experimentally impossible until recently. One of these new so-called next generation techniques now allows it to be done. And it's called long read sequencing. We've applied one of these techniques where you take RNA from human brain tissue and you try and keep the mRNA that's coding for the full length of the gene intact, and you sequence it from one end to the other, and you simply see whether you've got the whole of the gene left in the mRNA that's going to make the protein, or you've got exons missing. Okay. And what we did, and other people have found the same thing, is what actually is going on in human brain is not just a little bit different from hearts or from rats and mice. It is very different to the point where the things we have found are more different than similar. So although all species and all tissues have got the same gene, the way they're using it to make proteins, which is what really matters, is strikingly different. And that includes bits of the gene that we would predict would affect the binding of calcium channel blockers. Okay. Here's again the same cartoon. Perhaps the relevant bit is part B, where it says of all the hundreds and thousands of molecules of Catenin-1C we found in the brain, 40, well, almost exactly as many in grey, they had never been reported before. They didn't exist in the literature. They are different from the almost exactly the same number that were reported. So when you actually do it in human brain, things are 50% roughly different than what you thought they were going to be. Now, not all those differences are going to matter, but some of them probably do. And if you look at this region of the protein here, where we found about 13% of the RNAs encoding this protein were different than expected, that's the bit where the calcium channel blockers bind to. So it's at least possible, therefore, that in the human brain, you're making calcium channels that might differentially bind and be affected by calcium channel blockers in either direction. We don't know from our studies whether it's going to be better or worse, but it's different. So that's the general approach. You're identifying differences between brain and periphery. that affects the structure of the protein you're interested in, that then affects the ability of drugs to bind to that target. This is the last slide of this kind to show you. So in blue, we've got the brain, and red heart. And going down the list in increasingly light colors are the most abundant examples of CAT1C molecules. And the exons are in quite small numbers. But let me just point out, you'll see that many things are similar between brain and heart, but some are different. So for example, at this end of the gene, the heart always uses an exon called 1A. The brain always uses 1B. Further down the molecule, the brain always uses exon 22 and leaves out 21. The heart does the opposite. And there are many other examples too. So the bit of protein encoded by those exons will be different in the majority of brain and heart molecules. That's an example of how you might be able to use that information to tackle things differently. It's not just abundance that matters, though. You need to know much more about the biological differences between those isoforms. So, for example, some cells, this is on A is a picture of a human brain section, where some cells, as you can see, are stained red and some are blue, and those are different isoforms of the same gene, showing that different neurons choose to express one subtype or the other, or both. B shows that there's a risk for a polymorphism, a genetic variance, which affects the proportion of which of those isoforms you express. In C, you could do protein modeling to show that these differences affect the way the protein folds. And if they affect the way it folds, it affects the channel pore, therefore it affects how calcium can go through it and how calcium channel blockers drugs can work. And then in the bottom, there are different pharmacological and functional ways you can say, okay, if I put these different forms, the different isoforms of CAT1C into cells or animal models for that matter, what difference does it make to how much they flux calcium, how much they're blocked by calcium blocking drugs and so on? Okay. So the molecular work and genetics is just that gives you the evidence to bother to go into the lab and do what are really quite detailed and potentially expensive experiments. So, as I've said, you can, if you find these differences in isoforms, you need to know if they functionally make a difference. What you can then do is screen them against existing calcium channel drugs, blocking drugs, to see if those drugs affect these different forms of calcium channel in the same way. And as a separate work, you need to assess whether the drug you're interested in can get into the brain. What you could also do is compare these channels and screen them against drug libraries to identify new drugs, which might be much better at blocking the ones in the brain than the ones in the heart, because that's what you're really after. More efficacy in the brain and less side effects in the periphery. Okay. Now, all this stuff doesn't obviate in any way the need for clinical testing of these drugs. So as when it happens, you've still got the whole phase 1, phase 2, phase 3 pipeline to test them, but at least you'd be starting that process with, I think, a greater theoretical reason to think these drugs might be effective and probably safe, because we know in principle these sorts of drugs are. Okay, so that was a whistle-stop tour really through one of the examples, and there are many other pathways or targets I could have taken through to illustrate the points I'm trying to make. But just step back a bit and just again see how genomics can help us try and make better choices in discovering targets and drugs for psychiatric indications. First of all, genomics gives you evidence that target has got something to do with the pathophysiology of the condition. And if we're like the rest of medicine, that increases the chances that in the end you're going to get something that works. You can then use big data, if there are already drugs that target, to give you clues as to what might be possible. You can then drill in the same approach using existing drugs against that target to investigate it in more detail. If you think the target is or likely to be druggable, you can then do all the functional and pharmacological studies on the different forms, the different isoforms of the gene. And then lastly, you can feed in the pathophysiological evidence. You can pick pathways where it makes sense in terms of understanding the biochemistry and the neurobiology of that discovery, the disease. And then you go back and you do more discovery science. So as I said, I think the challenge is all this stuff takes time. This is real wet lab science. This isn't just done on computer predictions. And so unfortunately, geneticists can come up with more genes than you can ever study in the lab in a lifetime. And computational biologists will come all sorts of predictions about what should make a difference. But at the end of the day, I think you've still got to go back and you've got to demonstrate these things empirically. And that's where the time and the difficulties can arise. So my final slide. Cutting the chase, will psychiatric genetic discoveries lead to better treatments? And I perhaps predict I'm going to sit on the fence, probably, because it provides the rational basis for therapeutic targets. And as an aside, it's been the genetic discoveries that have really driven farmers' re-interest in our field. You'll all know that most of the big pharma pulled out of psychopharmacology research 10, 15 years ago. At least 2 or 3 of the big boys and girls are back in the field now. And rightly or wrongly, it's because they think the genomics is going to make the difference. It de-risks, they would say, to some extent, the whole endeavour. But, and again, I'm trying not to oversell this. I tried to emphasise it's really difficult to know what to pick. You've got so many genes that you just haven't got the time or the resources to investigate. The fact we don't have good models of psychiatric illnesses, and at least from research regards the need for access to human brain tissue, makes it a more difficult effort. I haven't talked at all about what you're trying to do. So I've assumed that calcium channels, if they're going to be a therapeutic target, you want antagonists, you want the blockers. But maybe in psychosis, the problem's the other way around. Maybe you want an agonist or you want an allosteric modulator. We don't really know the direction of effect of some of these genetic differences. I think that's an unlikely problem for the example I've given you, but in general, you find a gene associated with the illness, you have no idea what's wrong with that gene. Is it more of it? Is it gain of function, loss of function, or whatever? There's all other pieces of information, which again, you really want to know if you can. And as I said at the beginning, the small contribution of any one gene means it's a moot point how penetrant the therapeutic effects will be if all you're doing is tackling one gene. And lastly, I've emphasized that medicine shows us it can work, but let's remember in our own field, it hasn't been spectacular. Huntington's disease, we found the gene in 1983, I think it was. People have spent 40 years studying the biology of Huntington. And to my knowledge, they've still not come up with a treatment that can correct that abnormality. And likewise, ApoE, 1993, big effect on Alzheimer's disease, all sorts of clever cell biology. We still, to my knowledge, don't have an ApoE therapy for Alzheimer's disease. So in contrast, unfortunately, Ed's work, where cannabinoid will be in the clinic, I'm sure, within months, the genetic stuff is going to take a while. And I think the job of people like me is to tell you the potential without overselling it or telling you it's going to be a quick fix in your clinic, I'm afraid, in the next year or two. Anyway, thanks for your attention. I'm sorry if I've gone too fast.
Right, thank you very much. I feel like I can go and have my exam now, which I wasn't prepared to do so beforehand. It's obviously difficult for us with a clinical hat on really to really make sense really of that information. So I start from the very end of your talk. Is it really worth it, given that we've got very limited funding, to throw so much money really at this type of research, when it takes almost a lifetime really to get to a point where you can even start a trial into phase 1, 2, 3 sort of clinical?
I think that's a very appropriately skeptical view. I think if the only point of genetics was you thought it was going to lead to drugs, I would share that view. I think it's the fact that in a sense therapeutics is only one of the potential benefits. As I was saying, the Royal College is moving pretty close to it being helpful for the diagnosis of psychosis. So I think it's almost a free hit benefit on top of those other reasons for doing it. But no, I think the therapeutic benefits of genomics will come much later than the diagnostic or prognostic value of genomics. Yeah.
Okay. Comments?
Check if we've got any online.
Yes, please.
What is it that affects the splicing? Is it the mRNA or is there something upstream from that?
That's a great question. We don't know. There are lots of possibilities. So one theory is that actually it's not a master schizophrenia gene, but one of the mastery-like genes are genes that regulate the splicing events, independent of the individual.
How do you look for these then?
Well, we haven't, because that's a different form of research. But amongst the many schizophrenia genes, I think are some of the genes that are involved in splicing regulation.
Yeah. So what sort of research is that? A different research? Do you mean a different genetic type of research?
Well, I've never done genetics. We take the findings of genetics into the lab to do what you might call cell biology or biochemistry.
Yes.
The people who do the genetics and do the kind of clever statistical genetics to work out which of the genes and how do they group together that are contributing something inherited with, that's just a different field. Suffice to say, I don't think anyone's yet discovered things from that are clear enough for people like us to take them into account. And of course, it's not just genes that regulate splicing. It's the way genes are, for example, methylated or so-called epigenomics.
Sure.
They're environmental factors. So, I mean, it's a great question, but I don't know anyone who's actively studied.
So, what about the microRNA? People looking at microRNA, or you looked at long reads. Have they done long reads of the RNA, the microRNA, or the RNA?
Long read sequencing mostly still fairly targeted. So genome-wide long read sequencing is in its infancy. There's some big data there, but I don't think they're out yet. But you're right. I mean, one of the microRNAs is one of the schizophrenia genes, MIR137. So
Sure.
microRNA is part of it. Long coding RNAs are almost certainly part of it. It kind of makes my point. You just don't quite know what to start with. We focused on, if you like, the end of the process. But you're quite right. What drives that originally may or may not be genetic in the first place. And if it is, we don't know which gene it is that might be doing that. So yeah.
Coming back to another clinical perspective, what about the phenomenology really in terms of the phenotypes really of our patients' presentations? Do they help to some extent, and is there actually any perspective where there is an attempt to actually link that in with what we see in our clinical observations?
Again, that's not kind of what we do. But if you look at those big GWAS studies, where I think one I showed you has something like 75,000 patients in them, the price you pay for the sample size, you don't know very much about the phenomenology. You know they meet diagnostic criteria, you know some basics about them, but the possibility that some of the genes of schizophrenia are more to do with some subtypes than others, or symptoms than others, or cognitive dysfunction, I don't think we really know. I made the point that the calcium channels, like many other genes, they're transdiagnostic. So one view is, whatever they're doing something to the phenomenology that is common to these psychiatric disorders. They're unlikely to be contributing to first-rank symptoms, for example, because otherwise why are they equally robust associations of major depression? So then people say, what might that common phenotype be? And some people say it could be cognition. Some people say it could be sleep disturbance, working memory, because calcium channels are involved in those processes too. But again, how you distinguish between those questions is really difficult because we don't know enough about the patient, enough patients in detail about the phenomenon.
Okay. Any other questions? Yes, please.
Thanks for the talk. Multiple cancer channels where they're involved in lots of conditions, including cardiac and Lambert-Eaton, etc. Is there any overlap between these sort of these SNPs and their and the other conditions that help in, for example, finding out vulnerability or cardiac side effects of medication, or it could explain some of the cardiac effects that people
There's a little bit of evidence of that. So if you look at the equivalent GWAS, say, of cardiovascular disease, and you overlay that onto, say, the schizophrenia GWAS, there's a shared genetic predisposition to psychosis and to cardiovascular disease, partly explaining our well-known observations that our patients have high risk of those conditions. And the calcium channels are one of the gene families that are in that overlap. So yes, in principle, but again, the proportion of that overlap that they actually explain is tiny. But you're right, there are some rare variants and mutations in calcium channels that cause those much more penetrant cardiovascular phenotypes in Timothy syndrome, for example, where psychiatric aspect is quite prominent when it's there, but it's a very rare phenotype. It's not the kind of thing that you see in most patients most of the time.
One last question. In terms of, I mean, you probably have some kind of oversight as to how many labs are doing work on different of these targets. Is there anything in the pipeline that we will soon see within the clinical arena, at least at the clinical trial level?
Good question. Because some of the more exciting things that people keep most quiet, so confidential, so there may well be things that are further on than I think. I think I mentioned on my list above the calcium channels, there's a couple of the glutamate receptors, where because a number of companies and academic labs have been studying glutamate receptors anyway in schizophrenia because of all the other evidence that glutamate is kind of upstream of or complements dopamine. So those labs were further ahead, and then the genetics comes along and they can factor that in. So there's 2 or 3 glutamatergic drugs which are actually further down the translational pipeline. Whether anything will make it and whether that's to do with the genetic candidacy or it would have happened anyway, I don't know.
Okay, thank you very much again, Jim. So let me introduce you briefly. Our next speaker is Professor Anthony Grace, Distinguished Professor of Neuroscience, Professor of Psychiatry and Psychology at the University of Pittsburgh. He's also Editor-in-Chief at the International Journal of Neuropharmacology. And his research interests lie in the interface of neurobiology and psychiatry as well. He says on his website, our research interests lie at the interface in terms of experiments conducted in the laboratory, applying basic neuropharmacological approaches in the study of central dopaminergic systems. And obviously with the ultimate goal of determining the neurobiological correlates of mental disorders and some modes of action of psychotherapeutic drugs. So I'm very pleased to welcome you to our session this afternoon. I hope you can hear us well. And over to you, Anthony.
I can hear you well. Can you see my shared screen?
Yes, we can.
Okay, so what I'm going to be talking about is some of the work that we've been doing looking at novel therapeutic approaches to schizophrenia, as well as current therapeutic approaches and how they work. And I very much enjoyed listening to Paul Harrison, although I'm going to be talking about what we do think are effective animal models. So how can we model something as uniquely human as schizophrenia in an animal? Well, certainly we can't know whether rats are hearing voices or hearing squeaks. We can't get them to lie down on a couch and tell us about their mothers. But what we can do is take into consideration known risk factors for schizophrenia and go back and forth between clinical studies and basic science studies to cross-validate what we're doing. So to study this, what we've done is to take advantage of a known risk factor for schizophrenia. We know that schizophrenia is at least genetically associated, if not genetically driven. And we also know that the second trimester is a risk factor. So women who have severe influenza infections or in areas of famine or trauma during the second trimester have a higher incidence of schizophrenia births. So what we do is to get pregnant rats, and at gestational day 17, which is roughly equivalent to the human second trimester, we give them this DNA alkylating agent, MAM, methylase oxymethanyl acetate, and then examine them as adults. And when we do this, we see everything that you would expect in an animal model of schizophrenia. We see thinning of limbic cortical structures, the same limbic cortical structures that we see thinned in schizophrenia brains, but not a large loss of neurons. Instead, we lose neuropil and you get an increased cell packing density. We also find a selective loss of these parvalbumin GABAergic neurons in the prefrontal cortex and hippocampus in the model, as well as in human schizophrenia patients. We see a lot of consistent behavior things like sensory gating, like impairment of prepulse inhibition of startle, things related to prefrontal cortical function or executive function like reversal learning and extra-dimensional shift, which is sort of the rodent equivalent of the Wisconsin card sort test, as well as the negative symptoms, things that are missing from a normal personality, such as latent inhibition and impairments of social interaction. We also find consistencies in pharmacology, just like some studies that were done back in the 50s before ethics were invented, where they gave schizophrenia patients basically anything off the shelf. They saw that schizophrenia patients had a selective increased response to PCP, just as we see in the animal models, as well as an increased locomotor response to amphetamine that only emerges post-puberty. Now, this is consistent with amphetamine worsening psychosis, but of course, psychosis has to be present for it to be worsened. And finally, there are changes with respect to metabolic activity, especially hyperactivity in the limbic hippocampus. Scott Small and Scott Schobel have shown that there is hyperactivity in the anterior hippocampus of humans, which is the limbic hippocampus. I'll be talking about the ventral hippocampus in rodents, which is functionally equivalent to the anterior hippocampus in humans. So one thing, as I mentioned, that there's a big change in schizophrenia is the loss of these GABAergic parvalbumin neurons. In fact, there is a rather enormous loss on the order of 30% in schizophrenia brains post-mortem. And what we see in the MAM rats is the same thing, a decrease of parvalbumin-labeled GABA neurons in the ventral hippocampus and in the medial prefrontal cortex. This loss of parvalbumin neurons leads to hippocampal hyperactivity. So just like the metabolic imaging studies, we see that the neurons in the hippocampus of the MAM-treated rats examined as adults are firing much faster than in the control animals. Now, does this impact dopaminergic neuron activity? Because we know that there's a lot of evidence for a dopamine role in schizophrenia. Well, what we did is to look at 3 different parameters of dopaminergic neuron firing. Cells per tract, which is basically how many are firing, how fast they're firing, and their pattern. We look at these 3 parameters because we know that these are controlled by different circuits and have different impacts on behavior. And what we find in the MAM rats is a more than doubling in the number of neurons firing spontaneously without a change in firing rate or firing pattern. Now, this is important because a dopaminergic neuron has to fire in order to be responsive to behaviorally salient stimuli. So the more neurons that are firing, the bigger the response to any stimulus that comes in. And this is consistent with studies done by Oliver Howes and Philip Maguire in London, showing increased fluorodopa uptake. Fluorodopa is taken up into active dopaminergic terminals. So they see more terminals active. We see more dopaminergic neurons driving those terminals. So when the hippocampus is hyperactive, it can overdrive the ventral striatal nucleus accumbens area, which then inhibits the ventral pallidum. The ventral pallidum produces a very strong inhibition of the dopaminergic neurons. But when it's inhibited, you remove this inhibition and the entire population of dopaminergic neurons starts to become active and is now going to be responsive to stimuli that come into the system. This is what we propose to be the basis of the hyperdopaminergic state in schizophrenia. Now, how do D2 antagonists treat psychosis in schizophrenia? Well, whenever you give a dopamine antagonist, haloperidol, clozapine, any number of others, if you give them a single dose of the drug, you block dopamine D2 receptors. You then get a feedback excitation from the striatum, which increases the number of dopaminergic neurons firing, as well as their firing rate and burst firing as a compensatory mechanism. But if you administer the drugs over a period of 3 weeks or longer, what we find is that instead of an activation, we see a loss in the number of dopaminergic neurons firing. And this is not because they're inhibited. It's because the continued overexcitation from receptor blockade to feedback excitation to further blockade eventually drives the neurons into what we call depolarization block, where they're so hyperexcited that they inactivate their spike generating mechanism and cease firing, which of course is going to decrease dopaminergic release in the limbic areas. The induction of this depolarization block actually correlates very well with therapeutic efficacy and side effects. If we look at depolarization block in the ventral tegmental area, the area involved in schizophrenia, we find every drug that has antipsychotic efficacy also produces depolarization block in this area. In contrast, if we look in the substantia nigra, an area that's more involved in motor function, what we find are drugs that cause depolarization block in the substantia nigra tend to produce extrapyramidal side effects. And if we look at other compounds like metoclopramide or zacropide that have low therapeutic efficacy, don't produce depolarization block, but they do produce depolarization block in the substantia nigra and have a high propensity for producing extrapyramidal side effects. So we have this identity between, in the normal animal, producing these effects on the limbic system related to schizophrenia and the extrapyramidal system related to extrapyramidal side effects. However, in the MAM rats, we have a different initial condition. We don't have to worry about the increased number of dopaminergic neurons firing caused by the drug because they're already activated. So in the normal animal, the antipsychotic drug activates the dopamine system and then after weeks causes this depolarization block. But in the MAM rats, the neurons are already firing. All of the neurons are already active, which means now when you put this excitatory feedback stimulus of antipsychotic drugs on, you get a rapid induction of depolarization block that's persistent. And this really validates the need to use model rats, because this is something that we really didn't find looking at control animals. So in control rats, antipsychotic drugs first activate dopaminergic neuron firing to increase the number of dopaminergic neurons firing or population activity as a compensatory mechanism. And it takes weeks of treatment to overcome this. But in the MAM rats, the population activity is already maxed out. So the added drive leads to rapid induction of depolarization block. And this is actually consistent with the therapeutic picture. We know that the dopaminergic system is probably responsible for the positive or psychotic symptoms of schizophrenia. And it's been shown that the more positive or more psychotic symptoms, the faster the action of the antipsychotic drugs. In the MAM model, the more dopaminergic neurons that are firing in this model, the more rapid the antipsychotic drug causes this depolarization block, which then decreases the overdrive of the dopaminergic system. However, while the antipsychotic drugs are addressing this hyperdopaminergic induced psychosis, they don't really work at the site of pathology or on negative or cognitive symptoms. So currently, we think the site of pathology is in the hippocampus, where you have hippocampal overdrive from this parvalbumin neuron loss. But where we treat schizophrenia is by blocking dopamine receptors here, which is 1, 2, 3, 4, 5 synapses downstream from where we think the pathology is. Furthermore, if the hippocampus is hyperactive and dysrhythmic, it's also going to impact frontal cortical regions to cause cognitive deficits in the cognitive domain, and in the amygdala to cause deficits in the negative or affective state. But these symptoms aren't going to be touched by a drug that's working only on dopaminergic receptors. A better approach would be to treat at the site of pathology. And this is something that's been tried and unfortunately has failed in a number of conditions. One is Lilly's drug palmaglumitad, which is an mGluR23 agonist that looked really promising preclinically and looked really promising in initial clinical trials. But what they found is that it really didn't show an effective separation from placebo. When we look at this mGluR23 drug, studies by Beta Mogadam and others have shown when you look at normal animals, it really doesn't have much of an effect, either at baseline or when the dopamine system is stimulated. However, what about in the schizophrenia model? Well, what we find is that in our schizophrenia model, this palmaglumitad very powerfully reduced the number of dopaminergic neurons firing, whereas it didn't have this effect in the control animal. So choosing this 3 milligram dose, we wanted to see if we could actually get an effect if we gave it repeatedly. And what we found if we gave it repeatedly is the same effect as we saw acutely, a return to baseline in the number of dopaminergic neurons firing without a change in the control animals. We know that this is working through the hippocampus because if we inject the drug directly into the hippocampus, we get the same normalization of dopaminergic neuron firing only in the MAM rats. Now, in addition to treating the dopaminergic deficit, what we find is this drug is also effective in different other measures, like novel object recognition, which is something that's used to test cognitive potential. This is where we give a rat 2 similar objects, let them explore the similar objects, and then change one of the objects for a novel object after a 1-hour delay. Normal rats have no problem being able to make this identification. MAM rats don't really remember which is the novel object, but palmaglumitad restores this cognitive function. So the mGluR23 agonist can indirectly regulate dopaminergic neuron activity by the ventral hippocampus. And it's only observed in states of increased ventral hippocampal activity. Interestingly, it doesn't normalize dopaminergic neuron firing by causing depolarization block. It actually reverses the overexcitation. So this may be an effective target for ventrohippocampal pathology upstream of dopamine function. The inadequate clinical response that we see in the phase 3 and 4 trials, though, we think is due to another factor, and that I'll get into in a few minutes. Now, this is looking at something that's blocking some of the glutamatergic release. What about something that can actually normalize the hyperactivity of the hippocampal pyramidal neurons? Well, one novel drug that's come out from Neuron Pharmaceuticals is something called evenamide. Evenamide is a voltage-gated sodium channel blocker, but it works in a unique way in that it only tends to normalize hyperactive neurons. So low frequency firing neurons aren't really affected. High frequency firing neurons are very strongly inhibited. And what we find in the MAM rat is something very similar. No effect in the controls, but in the MAM rat, it decreases the neuron hyperactivity. And in fact, it does this by decreasing the hyperactive neurons and causing them to be more slower firing, moving them into the slower firing population. So in the hippocampus, evenamide is selectively inhibiting hyperactivity. When we look at the dopaminergic system, what we find is that evenamide very potently decreases the hyperdopaminergic state in the MAM rats. And interestingly, when we look at exactly which projections are affected, what we find is that evenamide is working primarily in the lateral VTA. Now, this is important because the lateral VTA projects to the associative striatum. And the associative striatum are where you tend to see the hyperdopaminergic state in schizophrenia patients in terms of amphetamine-induced raclopride displacement and fluorodopa uptake. So this seems to be normalizing dopaminergic activity precisely in the tegmental striatal area that's involved in psychosis. Interestingly, this effect persists well beyond the half-life of the drug, suggesting that the way this drug is working is by impacting plasticity more than just on baseline firing. And again, if we inject this drug directly into the ventral hippocampus, we also normalize dopaminergic neuron firing, suggesting that it's working in the hippocampus to normalize activity. Now, evenamide also works on novel object recognition. If we look at the two similar objects and then put a novel object, we find evenamide normalizes novel object recognition. And if we look at social approach, a measure of negative or affective state, where we have a rat with 3 chambers, a central chamber, 1 chamber that has a toy, and 1 that has a conspecific rat. What you find is that normal rats will tend to explore the cage with the rat rather than the toy. MAM rats don't discriminate, but after evenamide, this is reversed. So evenamide really has a unique action that doesn't depend on dopamine receptor blockade. The first and second generation antipsychotics block dopamine receptors. It compensates for dopamine hyperactivity, but works downstream from the site of dysfunction. And this, we think, leads to significant side effects, which underlie non-compliance and possibly treatment resistance. And this class of drugs are also ineffective in negative and cognitive symptoms, as well as treatment-resistant schizophrenia. By normalizing only hyperactive hippocampal neurons, evenamide seems to act at the site of dysfunction. So it can normalize activity without introducing significant side effects. And since it acts in the hippocampus, it can also normalize dysfunction, not only with regard to the dopamine system in psychosis, but also negative and cognitive symptoms. And by acting on the glutamate system, it can alleviate symptoms in patients that are treatment resistant. Since in treatment-resistant patients, there doesn't seem to be a lot of evidence for hyperdopaminergia in terms of fluorodopa uptake, but the glutamatergic system is still probably involved. And by attacking the glutamate system rather than the dopamine system, we're able to better normalize these effects. And the effects of evenamide, again, outlast its presence in the brain, suggesting it's having some long-term plasticity effects. Now, what about other approaches? We looked at normalizing the hippocampal system by acting on hippocampal pyramidal neuron hyperactivity. But the thing that drives this hyperactivity is a loss of parvalbumin neurons. So the parvalbumin GABAergic neurons release GABA onto the pyramidal neurons. These neurons are actually driven by glutamate acting on NMDA receptors. And this is probably where PCP can mimic or exacerbate schizophrenia. But the parvalbumin neurons release GABA onto a specific subtype of GABA receptor, the GABA receptor that contains an alpha-5 subunit. Now, the alpha subunit is where benzodiazepines bind. Normal benzodiazepines like Valium or diazepam bind at the alpha-1, alpha-2 subunit to relieve anxiety and cause sedation. The alpha-5 subunit is uniquely distributed just in the hippocampus and the amygdala, and it controls baseline firing activity. So what we were thinking is, if we're losing neurons, can we potentiate the effects of the neurons that are left by getting a benzodiazepine or a positive allosteric modulator that's selective for GABA-A alpha-5 receptors? And working with Jim Cook in Madison, Wisconsin, we got hold of a GABA-A alpha-5 benzodiazepine or PAM, positive allosteric modulator. When we give it systemically, we can very potently inhibit evoked activity in the ventral hippocampus. But if we look at a different glutamatergic synapse like thalamic evoked activation in the cingulate cortex, we don't get an effect. So it's selective for the hippocampus. Whenever we give this GABA-A alpha-5 drug, in the MAM rats and control rats, it has no effect. But in the MAM rats, it brings dopaminergic neuron firing right back down to baseline. Furthermore, we know it works in the hippocampus because if we inject it directly into the ventral hippocampus, where we have the parvalbumin neuron loss and hippocampal hyperactivity, it normalizes this activity. And whenever we look at amphetamine-induced hyperlocomotion, in the MAM rats, in saline rats, amphetamine increases locomotion. In the MAM rats, it causes a much greater increase in locomotion, but this alpha-5 compound brings the hyperlocomotion right back down to baseline. Now, a concern is that a number of drugs that showed promise in preclinical studies have failed in the clinic, including pomeglumitad, Roche's glycine uptake inhibitor, Pfizer's PDE10 inhibitor. All of these drugs looked really good preclinically, but failed in the clinic. And a number of drug companies took this as a rationale for leaving this space because they said, well, it's clear that you can't translate from animal models to the human condition. But we came up with a different interpretation, and that's that the pharmaceutical companies don't know how to run clinical trials. Now, why do I say this? Whenever we test these drugs in the animal models, the very first drug they see is our test compound. But in the phase 3 and 4 trials, they take schizophrenia patients that have been on antipsychotic drugs for years or even decades, withdraw them for 1 week in order to wash the drug out of the system, say the system is back to normal, and then test their compound. The thing is, the system isn't back to normal. Giving long-term D2 antagonists will cause dopamine supersensitivity. So not only do you have increased dopaminergic neuron firing, you have a supersensitive postsynaptic receptor. So what we wanted to test is to look at antipsychotic drug treatment and withdrawal in the MAM model. And what we found is that our GABA-A alpha-5 compound, which nicely normalized the hyperdopaminergic state in the MAM rat, after just 3 weeks of haloperidol and 1 week of withdrawal, it doesn't have an effect. Furthermore, when we look at hyperlocomotion, what we find is that the haloperidol prevents the alpha-5 PAM from having any effect on the hyperlocomotion. In fact, even lesioning the ventral hippocampus has no effect on the hyperlocomotion. So in the MAM rat, the normal hyperlocomotion that you're going to get in response to amphetamine that's normalized by the alpha-5 drug. After 3 weeks of haloperidol withdrawal, there's no effect. So haloperidol pretreatment altered the capacity of the system to respond to a novel agent. You're basically replacing the hippocampal overdriven dopaminergic neuron firing with a postsynaptic supersensitivity. And in those cases, once you withdraw a D2 antagonist, the only thing that's going to work is another D2 antagonist. Now, is there a way of circumventing the confounding actions of D2 antipsychotic drug pre-exposure in clinical trials? So Lilly showed actually in schizophrenia patients that patients at the early stages of the disorder responded better to palmaglutatide. And I actually was sat next to somebody in the audience who was working on some of the palmaglutatide trials. And what he said, there was one patient population that responded really well. And those were the patients that were non-compliant with their prior drug. But a better way, since we can't ethically discontinue medications for more than 1 to 2 weeks, again, we know there's nearly 70% non-compliance of patients with medications. So we could at first limit the studies to patients that can be verified to be long-term non-compliant. But another approach would be transitioning patients from prior antipsychotic medication that doesn't cause supersensitivity. Now, what drug can we use for this? Well, one drug that showed promise is aripiprazole. Aripiprazole, we found in the MAM rats, very potently normalizes dopaminergic neuron activity. Aripiprazole is a partial agonist, so it occupies dopamine receptors, and in fact, it's given at a dose that occupies them up to 95%, but it doesn't fully stimulate them. So it will stimulate them to a baseline level, but will prevent overstimulation. And what we find is aripiprazole, by its partial agonist action, actually directly inhibits dopaminergic neurons in the MAM rat without affecting the normal rat and without inducing depolarization block. So if we can give aripiprazole, if we can take patients that are on aripiprazole or Abilify, wash out the Abilify, and then test the novel compound, we might have a better model. Now, this is looking at drugs that are effective for treating schizophrenia. But we know that schizophrenia is bad for the brain. The longer the duration of untreated psychosis, the worse the prognosis. A better approach would be if we could do something that could actually prevent the transition to schizophrenia to begin with. And one factor that we know is involved in schizophrenia is stress. We know that stressful stimuli in patients with schizophrenia will exacerbate positive symptoms of the disorder. And we know that patients that are stabilized on antipsychotic medications that are returned to high stress environments tend to undergo relapse. But stress may also play a role in the etiology of schizophrenia. Eve Johnstone did a really interesting prospective study where she looked at children in Ireland that were at risk for schizophrenia based on family history, put them through a bunch of psychometric tests, and then followed them to see who converted and who didn't. And what she found are the children that were hyper-responsive to stress tended to be the ones that converted to schizophrenia later in life. And I remember talking to Ellen Sachs at a schizophrenia meeting about her experiences early on with schizophrenia. And she was saying that whenever she had a stressful experience as a child, her friends would get over it in about an hour, but it would persist for her for days. So this inability to regulate stress seems to be one of these important underlying factors. And stress, we know from Bruce McEwen and Sapolsky, can cause a loss of parvalbumin neurons in the hippocampus, which are the same neurons lost in patients with schizophrenia and that are lost in the MAM rats. So developmental disruption using gestational MAM also causes this effect. So if we look at MAM-treated rats pre-pubertally, before we see any of the changes in the dopamine system, any of the changes related to cognition or affect. What we find are the rats at this pre-morbid state are stressed, because if we look at the elevated plus maze, where we have 2 open arms and 2 closed arms, normal rats explore all the arms. When rats are anxious, they hide in the closed arms, which is what the MAM rats were doing. When we look at a measure of stress response, we look at foot shock vocalizations. This is where we give a mild foot shock. Whenever you give a mild foot shock, the animal freezes and splays out, but they're actually vocalizing ultrasonically. And the ultrasonic vocalization is a measure of the stress response. What we find in the MAM rats are way more vocalizations to the same stimulus that we see in the control animals. And finally, when we look at the amygdala, an area that we know is involved in stress and anxiety, in the MAM rat, it's hyperactive prepubertally. So if stress plays a role in the developmental etiology of schizophrenia, this suggests an early intervention in susceptible individuals may help to prevent this transition to psychosis. So we tested this in the MAM model by administering the anti-anxiety agent diazepam peripubertally and then testing the animals as adults. So we got a dose of diazepam, and then the MAM rats normalized the behavior in the elevated plus maze and brought amygdala neuron firing back to control. We then gave this drug to MAM or control rats 10 days before puberty, and then withdrew them and waited until they reached adulthood. So basically treating this anxious premorbid state. And what we found is that this was sufficient to blunt the loss of parvalbumin neurons and prevented the increase in the number of dopaminergic neurons firing, and just prevented what seemed to be this onset of the schizophrenia pathology by treating this early stress response. Now, of course, diazepam or Valium is not something that you're going to want to start giving to children. You don't want to put it in their frosted flakes. What about less invasive tools? Well, we heard about CBD and how effective CBD can be. And CBD is a good anti-anxiety agent. What we found is if we gave CBD during the same prepubertal period, what we found is it prevented the increase in the number of dopaminergic neurons firing, just like in the case of diazepam. So MAM animals showed an increase in dopaminergic neuron activity in the VTA, which was prevented by pretreatment with CBD. Now, this really gives us an interesting perspective. So converging evidence from clinical and basic science studies suggests inability to regulate stress early in life can lead to pathological changes in the hippocampus that may underlie the emergence of schizophrenia in the adult. But we think this is telling us something important, that the MAM isn't causing the schizophrenia in the rat. The MAM is causing hyperresponsivity to stress, and it's the hyperresponsivity to stress that leads to the schizophrenia phenotype in the MAM rats. And what I posit is we have the same thing in humans, where it's not the genes that are causing schizophrenia, it's the genes that are causing hyperresponsivity to environmental factors. And it's that hyperresponsivity to environmental stressors that lead to the pathology emerging later in life. And we know that this is consistent with a number of different factors. We know that even identical twins only show a 50% concordance with schizophrenia. So it's thought to be genetics plus some other factor. Furthermore, at least a third of schizophrenia is non-familial. And we know that childhood trauma alone is enough to increase the risk for schizophrenia later in adults. So if we can treat this stress effect, we should be able to prevent this transition. Now, I've talked about this using pharmacology, but if you take high-risk individuals, the rate of transition is actually very low. And starting to give drug treatment to people that may be at some risk, you're probably going to be treating a lot of patients that don't need to be treated. What about non-pharmacological approaches? Well, we tested this in the MAM rats by using environmental enrichment. What we did is we put rats in a larger cage with lots of toys to play with and more animals to interact with. And we did this for 2 weeks before puberty. And what we found is that this was enough to prevent the increase in the number of dopaminergic neurons firing. So what this suggests is if we can identify children that may be at risk based on family history or that are already showing abnormal anxiety responses as children and treat the stress, we might be able to prevent the transition later in life. Sort of something like Pat McGorry is doing with his Origin project in Australia, where he's taking at-risk youths and putting them into a safe environment, a safe interactive environment to try to mitigate some of these environmental stressors. So these data suggest that genetic predisposition combined with adolescent stress can lead to the emergence of schizophrenia later in life. If you have a high genetic predisposition, you may not need a lot of stress because the stress factors are being amplified in order to transition to schizophrenia. If you have a low genetic load, but a high prepubertal stress, you're likely to get the same kind of effect. And in fact, there's epidemiological evidence for this. We know that studies looking at Jamaicans that moved to the UK, when you look at the people from the Jamaica that moved to the UK, the adults may show some anxiety and depression and things like that, but their children show a 7-fold increase in the incidence of schizophrenia. Why? Because they are undergoing one of the biggest stressors, psychosocial stress, because they have to go into an environment where they're a minority and looked down upon and have to break into an established social order. In contrast, if you look at the barriers surrounding Sao Paulo, Brazil, where people are too poor to move and you have extended family support systems, the incidence of major psychiatric disorders is significantly less than the general population. So while treating at the site of pathology can be highly efficacious, the problem is the damage has already occurred. A more effective approach would be to treat stress in at-risk individuals early enough to prevent the transition to psychosis. And then just to thank all the people in my lab who did the work, Holly Moore, who brought the BAM model to our lab, Felipe Gomez, who did a lot of the work looking at the circuitry, along with Dan Lodge, Jason Zhu, who did a lot of the work looking at stress, along with Daniela Uliana, Susie Sonnenschein, who did a lot of the work with the novel antipsychotics, and our long-term collaborators, Margarita Behrens and Kim Do. And thank you for your attention. I'll be glad to answer any questions.
Thank you very much, Anthony. So that was a rich portfolio with, I think, a degree of clinical relevance. I suppose that particularly the latter part, really, I suppose, from a perspective of early detection or early prevention, I could see a number of interesting trials emerging from some of those interesting animal experiments. But obviously, it does beg the question as to whether we can at all make any use of the data from these animal trials. Really, can you just comment on that before we go into any detailed discussions? Is it really transferable?
I mean, I think that it's very transferable. The problem with the prior studies where they couldn't get efficacy on the drugs is they were using patients that had been treated for a long term with standard D2 antagonist drugs. And D2 antagonist drugs are really going to change the way that the system responds to the novel compounds. That's why a lot of these initial trials, the phase 1 and phase 2 trials, looked good because they were taking patients that were early on in the disease state. But when you start to look at the chronic patients, then you get the problems. just like with pomeglometad, when they did their secondary analysis, found that the earlier onset, people at earlier stages of their disease had the best response. And whenever we take the animals and treat them with these drugs, then we treat them with D2 antagonists, then we interfere with the novel compounds. And as far as the translatability, I mean, we're seeing consistency with respect to parvalbumin neuron loss, with respect to a hyperdopaminergic state, with respect to hippocampal hyperactivity, all things that have been shown in human schizophrenia patients that correlate with psychosis.
Okay, thank you. Questions, comments from the audience? And I suppose we can also bring back our first two speakers. I mean, you made an interesting comment about CBD for preventative purposes, if I understand that correctly.
Yes.
Any thing to add from your perspective there? Is that something that's somewhere trialed, explored as an option?
So one of the trials we're setting up at the moment is a CBD trial in clinical high risk. We're running it for 2 years. And yeah, we're looking at to prevent transition.
Yeah. And does genetics come into identifying at-risk patients in that trial?
Genetic?
Yeah.
Not so much, no, not anymore. It's more based on symptoms and presentation. Baseline. So I think a few years ago, there were people who had a history of psychosis and genetic risk. We recruit risk of developing schizophrenia is lower. So it's not such a useful group to target.
Okay. Yes, please.
Oh, thank you for interesting talk.
Can you hear?
I can't hear the questions, no.
Okay.
Sort of, what's your ideas about how to sort of, yeah. Apologies. Apologies for this way. I'm just, it's very interesting to think about the social factors, and I think clearly there's a lot of research showing that that's cases of interior like schizophrenia. Do you have any ideas how we could sort of, I'm sure we could actively help these children who are at risk, but sort of how to recruit them in this huge societies of ours?
So
Yeah, I'm sorry, I couldn't hear that at all. Can somebody?
okay. So I think the question is about how to identify at-risk subjects. I think, is that correct, Patty, that this is the main issue?
No, I mean, it's clearly you could help you pick them up.
So are we going by, I suppose, genetic risk factors in terms of stratifying risk, or are we essentially going by what is currently the clinical approach in terms of early detection services, which relies obviously on sort of, to some extent, predictors really coming from psychopathology? I guess that's still an open question, because, and I think then the next question is, where are these individuals presenting with those symptoms? Do we need to go into primary care or GP surgeries? Do we need to go into schools? Because I think it's an important question, obviously, in testing some of these approaches in at-risk patient groups.
Oh, absolutely. But I mean, I think that you can do it in a stratified way. First, when there's a strong family history of schizophrenia and depression, because we know that those do tend to run in the same families, to screen some of those children for their responses to mild stressors, such as a public speaking event or something along those lines. And the other is to alert teachers to look for children that seem to be anxious, especially the ones that are too anxious to have social interactions with other children. And identifying these people early on, testing them for stress responsivity, just something simple like galvanic skin response or baseline salivary cortisol or something along those lines, and just treat the stress. If they're stressed, you're going to have a positive effect whether they're going to get schizophrenia or not. But the ones that may be at risk for getting schizophrenia, you might be able to prevent that transition.
Which is an obvious case, obviously, for more psychosocial interventions as well. We're not only looking towards compounds, I suppose.
Absolutely.
Yeah. Can I ask you about evenamide? Where are we in the development of that drug in terms of testing for trials and coming to the markets?
So it's has gone through a phase 2 and phase 3, but it's being tested as an adjunct rather than as a monotherapy. And as an adjunct therapy, it seems to be pretty effective from the initial trials. I would love for it to be tested as a monotherapy, but it's really hard to get drug companies to test their drugs as monotherapies because they say if it fails as a monotherapy, it's hard to go back and use it as an adjunctive. So right now, as an adjunctive treatment, it seems to work quite well.
But it's not yet available. It's still in clinical trial phase, yeah?
Correct.
Yeah, okay. There's a question here from our online audience. Thank you for the interesting topic. Which measures in animal models, like elevated plus MACE, pre-pulse inhibition, might be candidate to better reflect the loss of GABAergic neurons and this hyper-responsive state in the context, in your opinion? So again, which measures in animal models might be candidate for to better reflect the loss of GABAergic neurons?
I mean, that's a challenging question because, of course, what we would like to see is a measure of psychosis, and we don't really have a good measure of psychosis in animals, except for things like distractibility, which could be cause for a lot of other things. That's why we rely on things like recording from the hippocampal to look at hippocampal activity and recording from dopaminergic neurons to look at dopamine activity. Because the hyperdopaminergic state, as far as dopaminergic neuron activity, seems to be a very good correlate with psychosis in humans and imaging studies in humans. For the loss of GABAergic neurons in rodents, probably the best way is to be looking at ventral hippocampal pyramidal neuron firing activity and just the number of neurons firing and how fast they're firing in the ventral hippocampus. Pre-pulse inhibition of startle is not really a good measure because you see pre-pulse inhibition of startle under so many pathological conditions in drug abuse, in anxiety, in dementia. So although it's something that's necessary, it's not really something that's selective. The best way to do is to use the combination of measures, looking at elevated plus MACE, looking at foot shock-induced vocalizations, looking at things like novel object recognition. And it's the confluence of all of these behavioral variables that seem to be the best measure, because that gets around pathologies that are going to affect one measure, but not the other measures, and probably isn't going to be a good measure of schizophrenia potential.
Okay. Can you say something about the use of benzodiazepines? Do you think that we underusing them in acute psychiatry in terms of a tool that drives down GABAergic activity, maybe also longitudinally, at the price, obviously, of creating addictive profiles?
I mean, I don't think that there's a schizophrenia patient out there that hasn't been exposed to benzodiazepines at some point. I mean, it's a great anti-anxiety agent in a disorder that has a lot of anxiety associated with it. There have been some studies done looking at benzodiazepines in ultra high risk patients. And it doesn't, although it seems to be good at controlling the anxiety, and even there have been some studies showing that it can actually decrease some of the psychosis even as a monotherapy, it doesn't prevent the transition because it's too late. You have to get it at the very early stages, the prepubertal, peripubertal stage. So children about 10 to 15 years of age that are going to be at most risk for stress-induced emergence of psychosis later in life.
Yeah. Right. Yes, please. I will translate it, yeah.
I think reflecting back, it's quite sad to hear that some of the cases who are on long-term olanzapine think their brain structure is so different that they actually don't benefit from these new drugs. So I guess two questions. One is, should we as prescribers be a lot more thoughtful about who we start people on? For example, should ibuprofen be first line? And then secondly, there's often a discussion that we don't go to clozapine quick enough. And have any of these experiments been done with clozapine as well?
Right, I'm trying to capture that question. So it's about a recognition of the fact that you described really how some of these drugs almost sort of block out the receptors to the point where the depolarization makes it very difficult for new agents to actually have any relevant effect. So the question was, A, should we move away from these drugs more and consider aripiprazole more for first line? And is there also an issue really in that respect to consider clozapine more first, second line, really, rather than only in treatment-resistant schizophrenia?
Well, I think the issue about using clozapine as a first-line drug has been something that's been discussed repeatedly over a number of years. Because on the one hand, it's very effective. On the other hand, it's sort of your last current line of defense, and you don't necessarily want to wear it out early. But I think starting out with clozapine is probably a good idea. It's still going to run into the issue of this depolarization block induced dopamine supersensitivity, which is a big problem. Aripiprazole is probably a better drug to use, but it doesn't have the efficacy that something like clozapine or even some of the other standard treatments have. But for patients who respond, it's probably a better drug to use, either that or brexpiprazole or any of the other partial agonist drugs. I mean, the problem with the D2 antagonists is that the supersensitivity they induce is rather long-lasting. And if you're going to try a novel compound, you're probably going to have to cross-titrate over a rather extended period of time. I mean, it's interesting whenever you look at people that have been on a standard antipsychotic drug and have transitioned to aripiprazole, the first thing they see with aripiprazole is a big exacerbation of the psychosis. Mm-hmm. And what I would say is that's the aripiprazole knocking them out of depolarization block. But then as they're knocked out of depolarization block, you get a lot of dopamine release, you get the normalization of the supersensitivity. So what I think we need to do is to get people transitioned onto a drug that's going to stabilize them without causing depolarization block long enough to get the system back to a baseline state. And once it's back to a baseline state, probably after a month to 6 weeks, then these novel compounds should be much more efficacious. But of course, the best would be to treat patients right as they come in with these novel compounds, especially once they're approved. And you know, the KarXT is one drug that doesn't work by blocking dopamine receptors, that it has been shown to be an effective antipsychotic drug. And that might be a better first line of defense than standard D2 antagonists.
Thank you very much. Does that answer your question? Good. Yes, please.
It's on the same topic.
Yeah.
Just like, how long would you use these? And if you didn't see a response, would you then switch to standard antipsychotics? So if you are saying try these first, say someone's admitted, try these first, and say there's no improvement after a week, 2 weeks, 3 weeks, 4 weeks, when do you then, how long would you expect it to take?
Yeah, I mean, this is a question also related to these new compounds, which we obviously not yet having in clinical practice. I mean, would you have any recommendation as to for how long patients should be exposed to those before then moving back to actually using traditional substances?
If they don't work.
If they don't work.
It depends on the treatment history of the patient. If you're talking about patients that have been on standard antipsychotic drugs for a very long period of time, it's going to take probably a month to 6 weeks to get the system normalized. But you don't want to have someone psychotic for that period of time. You know, this is really an empirical question that's going to need to be tested. And probably cross-titration is a much safer way to do it than stopping a drug, switching to another drug, seeing if it works, and then switching them back.
I was meaning more about first episode, really.
Okay.
So say you have a first
Yeah,
episode.
well, I mean,
presentation
we
talk.
don't know yet because these substances are not even in use, so.
If it's first episode, these drugs should have a rather rapid onset of action if they haven't been exposed to prior to D2 antagonist administration. So I would say if you don't get a response within the first week, then go back to other standard of care.
That's very helpful, thank you. Yes, please, Ed.
There's a lot of drugs that we need to go back and test again with better design trials as a result.
Yeah, the question is, I mean, we've moved obviously forward in terms of our trial design, really. Do you think there is a case for some of these drugs that have been previously regarded as non-effective to be retested with new design methodologies?
Absolutely. In our hands, palmaglumitad is a fabulous antipsychotic medication. Doesn't do anything to controls, very effective at treating the MAM animals as far as the hyperdopaminergic state, the cognitive deficits, and the emotional deficits. And like I said, I think this drug, if it had been tested in patients that hadn't been contaminated with years of D2 antagonist treatment, I think it would be a great drug. And even Lilly's, secondary analysis showed that people at early stages of the disease tended to respond much better. Conjectural things that people that were non-compliant with their prior medication tended to respond better. And we know that antipsychotic drugs have a 60 to 70% non-compliance rate. So if you can test patients for compliance, long-term compliance by hair samples or blood samples or things like that, get people that are verifiably non-compliant or that haven't been exposed to these drugs, I think these drugs are going to be very effective. And I believe that there is some work that's going on that's trying to sort of resurrect palmaglumitad as an antipsychotic drug with better clinical trials.
Thank you. Right, I think we probably exhausted all the questions. There's one more here online. Do you see there's evidence in very late onset schizophrenia with these novel drugs?
Evidence for
for these novel drugs in late onset schizophrenia? Any particular case for these drugs in late onset schizophrenia?
You mean to be used in late onset schizophrenia?
Yeah.
That's an interesting possibility. We don't really understand the pathophysiology of the late onset. We know that there is a late onset in women that hit menopause, and that's probably due to the protective effects of estrogen that eventually wear off, which may then expose them to the same pathophysiology. But I don't think enough studies have been done looking at the late onset to see whether the metabolic changes that occur with the late onset are the same with standard treatment, standard schizophrenia.
Okay, I think we can bring the session to a close. Really big thanks again to our three speakers. And I think we've learned a lot sorry, you wanted to say something, Shiraz? The social after. Yes, I was just reminded to remind the people with the social after. Unfortunately, we can't invite you for drinks after, Anthony, but next time in London. Thank you.
Okay.
Thank you very much.
Thank you.